The last thing that we'll be discussing with regards to gene regulation, or the regulation of gene expression, is the role of activator and repressor proteins. Regulatory molecules, as we've said, sometimes interact with the outside of the double helix. That's an epigenetic control mechanism. And the kinds of proteins that do that work are called transcription factors. Sometimes, though, regulatory molecules actually interact with the gene itself. So we're talking about the time after the DNA strands have been separated from each other, after the gene itself is exposed, when the RNA polymerase enzyme would come in and begin this process of transcription. Remember, there are protein coding areas on a gene, and then there are regulatory areas on a gene. So activator proteins and repressor proteins are going to come in at this point and literally bind to the regulatory regions of the gene in order to control the transcription of that gene, in order to either upregulate transcription or downregulate transcription. Now, repressor proteins will come in and bind in the regulatory region to a place called an operator site or an operator sequence, a little region of the gene where the repressor protein can come in and sort of land, sort of bind. Okay? Repressor proteins, like the name suggests, are regulatory proteins that are going to down-regulate transcription. So when they come in and they bind to these operator sites, they are going to physically block the ability of RNA polymerase to come in and land on the promoter and start transcription. Repressor proteins bind to operator sites and block the RNA polymerase enzyme from being able to transcribe a gene. Activator proteins are essentially the opposite. Activator proteins also bind to the regulatory region of a gene, but they're going to bind to activator sites, just little places in the regulatory region of a gene that are called activator sites. When activator proteins bind to activator sites, they will actually make it easier for the RNA polymerase enzyme to come in and land on the promoter. They're going to actually enhance the ability of the RNA polymerase enzyme to land on and bind to the promoter and begin transcription. All right, now activator proteins and repressor proteins don't work alone. In other words, something has to set them in motion. And that something is often the product of what that gene makes, if that makes sense. So some genes, as we've said, are operons, and they code for a whole set of enzymes that are going to do a metabolic process for the cell. They're either going to build something in an anabolic reaction, or they're going to break something down in a catabolic reaction. So the products of those metabolic processes are what set these repressors and activators in motion. For example, there is a type of carbohydrate in microbial cells called maltose. We know it's a carbohydrate, remember, because it says OSE at the end of the name. So it turns out that there is a maltose operon. It is a gene, it's one gene that codes for several proteins. And those several proteins are enzymes. And those enzymes are what is needed by the cell to metabolize maltose, to break down the maltose sugar. So let's imagine a bacterial cell that prefers maltose. It likes to use the maltose sugar for energy. So if maltose is around and maltose is available, then the cell is going to want to make the enzymes that will break down that maltose so it can use it for energy. So when maltose is around, in other words, the cell wants to upregulate the gene, the operon, that makes those enzymes. So it turns out that maltose itself will bind to an activator protein. That activator protein will then change shape a little bit, and then it will be able to bind to the activator site of the operon, and then it will upregulate the transcription of that operon, and the cell will get the enzymes it needs to break down all that maltose. Okay, make sense? Let's take a look at a diagram of this process. Up at the top of the slide, we're shown a gene, and this happens to be an operon. It's a gene that codes for several proteins. So in blue and in yellow, we're looking at regulatory regions of this gene. And in green, we're looking at structural genes, structural or protein coding portions of the gene. So they've got these labeled MAL-E and MAL-F and MAL-G. These are 3 different codes for 3 different proteins, 3 enzymes. And these are the enzymes that are needed for the process that breaks down the maltose sugar. Now, if there's no maltose in the cell right now, there is no need for the cell to transcribe and translate this operon. It doesn't need to have these enzymes around if there's no maltose for them to break down. So it makes sense that when there's no maltose available in the cell, this operon will not get expressed. But now let's imagine that there's lots of maltose around, and they've drawn the maltose as this little blue circle. All right. Normally, again, no maltose. Well, the gene just doesn't get transcribed. The RNA polymerase enzyme doesn't attach to the promoter, and the activator protein doesn't help, and we get no transcription. But now let's imagine that maltose is around. They've drawn the maltose as this little blue circle, and they've labeled it as an inducer. The presence of maltose is going to induce the transcription of this gene. And how it works here is that the maltose will bind to the activator protein and cause the activator protein to change shape. And when it changes shape, it's able to bind to the activator site, shown in blue. So now this protein is actually sticking on the activator site of this gene. And by doing that, it makes it easier for the RNA polymerase enzyme to come in and bind to the promoter. And remember, the polymerase enzyme has to bind to the promoter in order for transcription to begin. So here we have the molecule that is involved with this gene, the maltose, acting as an inducement to turn the gene up, to upregulate the gene. And transcription will proceed. We're going to transcribe this operon. We're going to translate these 3 proteins. We're going to get these 3 enzymes made, and those enzymes will then metabolize or break down the maltose. Now, I also want to give you an example of a repressor protein and an operator site and downregulation of an operon. So let's talk about the Arginine operon for a minute. This operon contains several protein coding regions that code for enzymes that are going to build arginine. Now, all arginine is an amino acid, okay? So these are enzymes that are going to help an anabolic reaction occur that builds arginine. Now, let's imagine a situation where a bacterial cell has lots of arginine available. And remember, the cell is going to use that arginine and other amino acids to make protein. So there's plenty of arginine available in that cell right now. So that means that the cell does not need to transcribe and translate this operon. It doesn't need to make those enzymes because it doesn't need to build any more arginine right now. It has arginine. So the way this works is the arginine, again, the arginine itself is going to bind to a repressor protein. That's going to make the repressor protein change shape a little bit, and then it will be able to bind to the regulatory region of the arginine operon. It will bind to what's called the operator site. And when it does that, it will block the RNA polymerase enzyme from binding to the promoter. So it will downregulate the transcription of the arginine operon. And here's a drawing of that process. So up at the top of the slide, we're shown the arginine operon. Here's the promoter site. Here is that regulatory operator site. And here are the structural genes. Again, there's 1, 2, 3 proteins coded for in this operon. And these are enzymes. These are the 3 enzymes that are needed for the anabolic process that builds arginine. Here's the repressor protein down here in red. Now, under normal situation, this operon gets transcribed and translated. Remember, we said at the beginning of this lecture that the default position for most of these genes is go ahead and transcribe them. And that's what's happening. Here, this RNA polymerase enzyme is binding to the promoter, and now it's going to go ahead and transcribe these structural genes so that we make these enzymes and we build some arginine. But now we're going to imagine the situation where there's plenty of arginine around. And they've drawn the arginine as this little pink ball this time. Now you'll notice they have it labeled as co-repressor. Don't worry about that term. Just know that it's the arginine itself that is going to come in and bind to this red repressor protein. And when it binds, it's going to make the repressor protein change shape. That's going to allow it to bind to the operator region. And it literally acts like a wall. It acts like a block. It's an obstruction. Because now, even though the RNA polymerase enzyme can still bind to the promoter, it can't move forward. It cannot physically travel down this gene and transcribe those enzyme proteins. It can't do it because this is in the way. So when there's plenty of arginine around, transcription gets downregulated. We don't need those enzymes right now. We don't need to make any more arginine right now because we have arginine. We'll finish up today with a summary of what we've discussed about the regulation of gene expression. Remember, microbial cells must regulate most of their genes. They have to regulate the expression of those genes so that they're not wasting time and energy and resources making proteins that they don't need. The regulation can occur at several different locations, from transcription through translation to the production of active proteins. Cells can step in at any of those time points and control whether or not active protein gets made. Regulating at the level of transcription is the most efficient way to regulate gene expression because it prevents the wasting of materials and energy. It prevents time and energy and resources from being wasted doing any more of this process than is necessary. When cells control gene expression by controlling transcription, they do it in 3 areas. They do it with the state of their chromatin. That's an epigenetic mechanism. They do it with proteins called transcription factors. That's also an epigenetic mechanism. And they do it with activator and repressor proteins that interact with the gene itself. Remember that unlike eukaryotes, prokaryotic organisms have some genes that are called operons. They cluster certain protein coding portions of genes together into a single gene under the control of one regulatory region. These genes are related to each other. They're going to produce proteins that are related to each other. For example, they're often enzymes that are involved in the same metabolic pathway. Metabolic products are often used in microbial cells as regulatory molecules. For example, the maltose we talked about and the arginine we talked about. Those metabolic products are going to control activator proteins and repressor proteins, and therefore control the transcription of the operon. Repressor proteins, remember, will bind to operator sites in the regulatory region of an operon, and those will essentially block or downregulate gene expression. Activator proteins are different. Activator proteins bind to activator sites in the regulatory region of an operon, and they will stimulate or upregulate gene expression.